Welcome back to the Deep Dive. Today, we're diving headfirst, really getting into the nitty-gritty of biology. The cell.
That's right. It's the absolute foundation.
It really is. The blueprint, the command center for everything you are. And we're working entirely from the source material you sent over, ready to peel back all those layers.
And the goal today, it's not just about, spitting out definitions. We really want to make sense of these diagrams, these texts, turn them into something practical.
Functional knowledge.
Exactly. We're decoding the cellula, what the sources call the fundamental unit of life. We'll look closely at its machinery, how it works dynamically, and, this is crucial, how tiny events inside this microscopic world translate into big things.
Like moving, or even thinking.
Precisely. Movement, thought, how your body processes energy, it all starts here.
Okay, so let's map this out. We usually think, organ systems, but we need to start smaller, much smaller. The sources actually place the cell very specifically in the hierarchy.
They do. If you picture that hierarchy, like in Figure 4 they provided, the cell is right there at the fifth level.
Fifth level.
Yeah, you start super small with atoms. They build up into molecules, then bigger ones, macromolecules. Then you get the organites, the little structures inside the cell.
Ah, the subcellular bits.
Right. And then you assemble all that into the complete working cell.
So it's sitting above those internal parts, but it's the building block for the next level up, the tissues.
Perfectly put. It's the master building block. It holds that title. The basic morphological, functional, and genetic unit of living matter.
That means everything that defines life.
Pretty much. Metabolism, breaking down food, building things up, responding to signals, passing on genetic information, it all happens within or is controlled by this single unit.
So to get how it works, we need to explore its different zones, territories.
Good way to think about it. We've got the outer border, the membrane, then the bustling inner landscape, the cytoplasm, and finally, the control room, the nucleus.
Let's start right at the edge then. The barrier that defines the whole thing, the cell membrane.
Okay, the cell membrane, also called the plasmalemma. It's way more than just a bag holding everything in.
It's active.
Very. It dictates the cell's shape, for one, and it's the gatekeeper, constantly deciding what gets in and what stays out, separating the controlled inside from the unpredictable outside. The structure itself is fascinating. It's described by the fluid mosaic model.
Fluid mosaic. That sounds, fluid, not rigid, and complex, like a mosaic.
Exactly. It implies movement, components shifting around. The core structure, based on the sources, is mainly two things: phospholipids and proteins.
Lipids and proteins, fat and machinery.
Sort of, yeah. Think of it like a thick, oily sea. That's the lipids, and floating in that sea are various machines, the proteins. They're not fixed in place. They can move laterally, hence the fluid part, like icebergs in a fatty ocean, as the model suggests.
And the phospholipids, they form that famous bilayer. Looking at Figure 5, it seems like their basic nature is the key to how the barrier works.
Absolutely critical. Each phospholipid molecule is what we call amphipathic.
Meaning?
Meaning it has two distinct personalities towards water, a part that loves water and a part that hates it. In the diagram, see component 4A. Those are the hydrophilic heads. Hydro water, philic loving.
Okay, 4A, the heads, they like water.
Right. So they naturally turn outwards, facing the watery environment outside the cell, that's label 1, the extracellular space, and inwards, facing the watery inside of the cell, label 2, the intracellular space.
Makes sense. And the other part, component 4B, the tails.
Ah, those are the hydrophobic tails. Hydrowater, phobic fuene. These are long fatty acid chains, and they hate water. So what do? They turn away from the water, tucking themselves into the middle, facing each other.
Back to back, forming the core.
Exactly. They form this thick, oily, water-repelling layer right in the center of the membrane. This hydrophobic core is the fundamental barrier. It's what stops most water-soluble things from just drifting through.
So this water-resistant middle is the key to control. But mosaic implies more than just the lipid background, those proteins.
Yes, the proteins. Labeled 3 in Figure 5. These are the functional units embedded in the lipid sea. Some go all the way through transmembrane proteins, others are just attached to one surface or the other.
And they do specific jobs.
Crucial jobs, especially transport. They act as channels or carriers, like controlled doorways, letting specific molecules cross that hydrophobic barrier that they couldn't cross on their own.
Okay, so lipids form the basic barrier. Proteins provide the specific gates. The sources also mention a couple of other things in Figure 5. Cholesterol and some attachments on the outside.
Right. Cholesterol, labeled 5, slips in between the phospholipid tails. You might think cholesterol is just bad, but here it's vital. It modulates fluidity.
How so?
It stops the tails from packing too tightly together when it's cold, preventing the membrane from becoming too rigid. And it limits excessive movement when it's warm, stopping it from becoming too fluid. It basically keeps the membrane at the right consistency for the proteins to work properly.
A fluidity buffer, a stabilizer.
Precisely. And then on the outside surface, you see those branching structures. Those are glycolipids, 6, and glycoproteins, the sources call them glucids, 7, attached to proteins. Basically, carbohydrates linked to lipids or proteins.
What's their role?
They often form a layer called the glycocalyx. And a key detail from the sources is that these carbohydrate chains often carry a strong negative charge. They're super important for cell recognition, adhesion, acting like the cell's ID tags, how cells know who their neighbors are, or identify invaders.
Okay, so this whole structure creates selective permeability. It's not just theory, it affects everything, every day. If I'm a molecule, say oxygen, trying to be in...
Your experience, it totally depends on who you are chemically. Are you fatty or watery? Big or small? Charged or neutral? The hydrophobic core, that oily middle layer, is a major roadblock for anything with an electrical charge, like ions, sodium, potassium, calcium. Forget it, they can't cross on their own. Same for most water-soluble molecules, like sugars or amino acids. The barrier just rejects them.
But if you match the core's nature, dissolves like...
Exactly. If you're nonpolar, fat-soluble, you get a free pass. You just diffuse right through. Think about oxygen, text O2. It's small, nonpolar. It has to get in easily to fuel the mitochondria.
For energy production.
Right. And carbon dioxide, text O2 too, the waste product, it's also small and nonpolar enough to diffuse out easily. Another example, steroid hormones. They're derived from cholesterol, so they're lipids. They slip right through the membrane to find their receptors inside the cell.
The sources also mentioned that some small, uncharged polar molecules can sneak through too, like urea or ethanol.
Correct. They aren't perfectly lipid-soluble, but they're small enough and lack a strong charge, so they can manage to diffuse across maybe just a bit slower than pure lipids.
But the big important stuff often is charged, or polar. Glucose, for instance, the cell's main fuel.
Glucose. Prime example. It's relatively large and it's polar, water-soluble. It hits that hydrophobic core and bounces right off. No entry.
So it's stuck outside.
Without help, yes. Glucose, amino acids, ions like sodium and potassium that are vital for nerve function. They all need those specialized protein transporters we mentioned earlier. They need a specific mechanism to get across. This selective permeability, this control over traffic, is absolutely fundamental to life.
The membrane, a very smart, very selective border guard. Okay, we've secured the frontier. Let's venture inside into the inner landscape, the cytoplasm.
Right, the cytoplasm. This isn't just empty space inside the membrane. It's basically everything within the cell except the nucleus. And it's incredibly busy, a highly organized, complex environment.
But the sources describe it as a colloidal system. What does that actually mean?
It means it's not just simple salty water, it's a gel-like substance where lots of large molecules and particles are suspended. It has structure, different consistencies in different areas. This organization is important because it helps control where and when complex chemical reactions happen. Things aren't just randomly bumping into each other.
And it's divided broadly into two main parts, the background goo and the functional machines.
That's a good way to put it. The background goo is the hyaloplasm. That's the unstructured watery part, the solvent where many basic metabolic processes, like the initial breakdown of glucose, start. The functional machines floating within it are the structured parts, the cellular organites, or organelles, as they're often called.
Let's look at those machines, the standard toolkit almost every cell has. We can use figure 6 as a reference. First up, you need to build stuff, specifically proteins.
That happens at the ribosomes. You can see them as tiny dots, labeled as component 9 in figure 6. Sometimes called the corpuscles of Palade after the scientist who discovered them. These are the protein factories, period.
Factories.
Absolutely. Whether the cell needs an enzyme to speed up a reaction, a hormone to send a signal, or structural proteins to build itself, the actual assembly line is the ribosome. It reads the genetic instructions coming from the nucleus and stitches amino acids together in the correct order.
Okay, proteins assembled. Now, power, energy. That's the job of the famous mitochondria, component 3, the powerhouse.
The powerhouse, indeed. You can't overstate their importance. This is where the main energy extraction happens, through a process called oxidative phosphorylation. They take fuel molecules, like the products from breaking down glucose, combine them with oxygen.
The oxygen that diffused across the membrane earlier.
Exactly. And through a complex series of reactions, they release a huge amount of energy, capturing it in the form of ATP adenosine triphosphate. That's the cell's usable energy currency.
And the number of mitochondria tells you something about the cell.
Definitely. A cell that does a lot of work, like a muscle cell constantly contracting or a nerve cell firing signals, will be packed with mitochondria. A less active cell, maybe like a fat storage cell, will have fewer. It's a direct measure of metabolic activity.
That ATP production must be relentless.
Oh, absolutely. And think about this. Remember that sodium-potassium pump we mentioned? The one maintaining the membrane potential?
Yeah, the 3 sodium out, 2 potassium in pump.
That pump alone can burn through something like 20% to 40% of all the ATP the cell makes, just maintaining that basic electrical readiness.
Wow, that's a huge energy cost just to sit there ready.
It tells you how critical that ion balance is, and the mitochondria are working constantly to meet that demand, among many others.
Okay, incredible. So we have proteins being made, energy being generated. How does this stuff get moved around inside the cell? That sounds like the job of the endoplasmic reticulum, the RE components 7 and 10 in the diagram.
That's its main role. The RE is like the cell's internal highway system and manufacturing plant combined. It's a vast network of interconnected membranes forming sacs and tubules that spread throughout the cytoplasm. And it comes in 2 flavors, rough RE and smooth RE.
Rough and smooth. Component 7 is labeled rough, and 10 is smooth. What makes them different? Ribosomes again?
Exactly. The rough endoplasmic reticulum, RER, or ergastoplasm as the sources also call it, component 7, looks rough under the microscope because its surface is studded with ribosomes.
The protein factors we just talked about.
Right. So the rough R is heavily involved in making proteins that are destined for specific locations, either to be secreted out of the cell, inserted into the cell membrane itself, or delivered to certain organelles like lysosomes. It's also where proteins start to get folded into their correct 3D shapes. Quality control happens here.
And the smooth endoplasmic reticulum, SER, component 10. No ribosomes, so different jobs.
Correct. No ribosome, so it looks smooth. Its functions are more diverse. It's crucial for synthesizing lipids, including steroids. And as the sources specifically highlight, it plays a key role in glycogen metabolism, breaking down stored glucose, especially important in liver and muscle cells. It also helps detoxify certain drugs and poisons.
So rough RE proteins for exporter membranes. Smooth RE lipids, glycogen detox.
Got it. After things are made in the RE, where do they go for final processing and shipping?
That's the job of the Golgi apparatus, or Golgi complex. Component 1 in Figure 6. Think of it as the cell's main post office, or finishing and packaging center.
Post office. I like that. Yeah. Proteins and lipids arrive from the RE, usually in small membrane bubbles called vesicles. The Golgi modifies them further, maybe adding a sugar chain or trimming a piece off, then it sorts them, concentrates them, and packages them into new vesicles for their final destination. It's absolutely central for secretion, getting stuff ready to be shipped out of the cell.
Okay, manufacturing, energy, transport, packaging.
Hmm.
What about cleanup? Waste disposal.
That falls to the lysosomes, component 4. These are small membrane-bound sacs filled with powerful digestive enzymes, specifically hydrolytic enzymes.
Hydrolytic, meaning they use water to break things down.
Precisely. They can break down almost any large biological molecule, proteins, fats, carbohydrates, nucleic acids. Their job is intracellular digestion. They fuse with vesicles containing stuff brought in from outside, like bacteria engulfed by the cell, or they digest old, worn-out organelles from inside the cell itself, a process called autophagy, like self-eating for recycling.
The cell's recycling center and garbage disposal, all in one. Very efficient.
Extremely. Keeps the cell tidy and reclaims building blocks.
Right. One more major component shown in Figure 6 that seems universal. The centrosome, component 12. Looks important for division.
It is. The centrosome is the main organizing center for the cell's internal skeleton, the microtubules. And crucially, it plays a pivotal role during cell division, mitosis. It helps organize the mitotic spindle, the structure that pulls the duplicated chromosomes apart into the 2 new daughter cells.
So essential for reproduction of the cell.
Absolutely. Its presence is usually a sign that a cell can divide, which leads to a really interesting point mentioned in your sources.
Ah, yes, the exception.
Yeah.
Mature neurons lack centrosomes.
Right. And that's not an accident. It directly relates to the fact that most mature neurons lose the ability to divide. They become postmitotic. They're highly specialized for signaling, and they have essentially traded the ability to replicate for that specialized function and stability. No centrosome, generally no division.
Which is why brain and nerve damage can be so permanent.
It's a major factor, yes. Regeneration is very limited.
That perfectly illustrates specialization. The sources also mention other organites that aren't universal, but appear only in certain cell types for specific jobs.
Exactly. We've covered the core toolkit. Now for the specialist tools. For instance, muscle cells or muscle fibers are packed with myofibrils.
These aren't shown as a numbered component in Figure 6, but they're mentioned.
Right, they are the contractile machinery itself. Bundles of specialized proteins, primarily actin and myosin, arranged in a way that lets them slide past each other. This sliding movement, powered by ATP, is what generates force and causes muscle contraction. When you flex your arm, that's billions of myofibrils shortening inside your muscle cells.
Directly linking the microscopic structure to macroscopic movement.
Cool.
What about nerve cells? They have specialized internal structures too.
They do. Neurons contain neurofibrils. These are like internal scaffolding, bundles of protein filaments that help maintain the neuron's shape, especially the long axon, and might play a role in transport within the cell. They also have Nissl bodies.
Nissl bodies. The sources say these are basically the nerve cell's version of the ergastoplasm.
Exactly. Nissl bodies are essentially dense clusters of rough endoplasmic reticulum and free ribosomes found in the cell body and dendrites of neurons. Their presence indicates that neurons are protein synthesis powerhouses, constantly making neurotransmitters and proteins needed to maintain their structure and function.
So myofibrils for movement, neurofibrils and Nissl bodies for nerve structure and signaling. What about temporary stuff in the cytoplasm?
Yes, temporary inclusions, like component 14 shown in the diagram, often labeled as secretion granules. These aren't permanent organelles, they're just stuff the cell is storing or has produced. Could be droplets of fat, lipids, granules of glycogen, stored glucose, pigments giving the cell color, or vesicles full of products waiting to be secreted. They appear and disappear depending on the cell's activity and needs.
Okay, so the cytoplasm is this incredibly dynamic, structured soup filled with universal factories, specialist tools, and temporary storage. We've mapped the border, explored the factory floor. Now it's time to visit the head office, the command center, the nucleus.
The nucleus. Component 11 in Figure 6. This is usually the largest organelle, and it really is the cell's control center. It has three main jobs, according to the sources.
Okay, what are they?
First, it coordinates all the fundamental life processes of the cell. Second, it controls the cell's overall metabolism by deciding which proteins, especially enzymes, are made and when. And third, arguably the most critical, it stores the cell's genetic information, the DNA, and transmits it accurately during cell division.
The master blueprint and the manager. What are the key parts inside?
Well, first, it has its own border, the nuclear membrane or nuclear envelope, labeled 2. It's actually a double membrane, controlling what moves between the nucleus and the cytoplasm. Then inside, you have the karyoplasm, which is like the cytoplasm of the nucleus, a gel-like substance.
And floating in the karyoplasm is the most important stuff.
The genetic material itself. In a non-dividing cell, it exists as chromatin, labeled 5. This looks like a tangled mass of threads, but it's actually DNA tightly wound around proteins. When the cell gets ready to divide, this chromatin condenses, becoming shorter and thicker, forming the visible chromosomes.
Okay, membrane, karyoplasm, chromatin, chromosomes. And Figure 6 also shows component 13, the nucleoli, plural of nucleolus.
Exactly, nucleolus. These are dense little regions within the nucleus, not bound by a membrane. Their main job is to synthesize ribosomal RNA, or RNA, and assemble ribosomes.
So the nucleus makes the factories, ribosomes, that then go out into the cytoplasm to make the proteins based on instructions from the nucleus, a neat loop.
It's a very elegant system of control.
Now, usually it's one nucleus per cell, right? Mononucleated. But biology loves variety.
It certainly does. While most cells have one nucleus, there are exceptions. Your sources mention binucleated cells having 2 nuclei. A common example is hepatocytes, the main liver cells.
Why would liver cells need 2?
Liver cells are metabolic workhorses, doing a huge amount of synthesis and detoxification. Having 2 nuclei presumably provides double the genetic template, allowing for higher rates of protein production to meet those demands.
Makes sense. And then there are cells with even more.
Yes, polynucleated or multinucleated cells. The classic example mentioned is the striated muscle fiber. These cells are enormous, very long and cylindrical. Having multiple nuclei spread out along their length allows for efficient control and protein synthesis over that entire large volume. One nucleus just couldn't manage it all.
So number of nuclei relates to the cell's jod and size. And then there's the ultimate exception, the cell that throws its nucleus away entirely.
The adult erythrocyte, our red blood cell. As the sources clearly state, it's enucleated. It starts with the nucleus during development, but ejects it before entering circulation.
Why do that? Seems risky.
It's a trade-off. By getting rid of the nucleus and other organelles like mitochondria, the red blood cell maximizes the internal space available for hemoglobin, the protein that carries oxygen. It becomes an incredibly efficient oxygen delivery sac, but it pays the price. It can't make new proteins, can't repair itself, and can't divide. It has a limited lifespan.
A remarkable specialization. Sacrifice everything for the mission.
Well,
Right, we've got the structure down, the membrane, the cytoplasm, the nucleus. Now let's shift focus to how the cell lives and acts, how it interacts, moves things, uses energy, communicates. Let's go back to the membrane's role as gatekeepers and look closer at transmembrane transport.
right. How stuff actually gets across. The big divide here is energy. Does it cost the cell ATP or not?
Let's start with the free ride, passive transport.
Passive means no ATP required. The driving force is simply the concentration gradient, the difference in the amount of a substance on one side of the membrane versus the other. Things naturally move from where they are more concentrated to where they are less concentrated, downhill, energetically speaking.
We already talked about simple diffusion, like oxygen just slipping through. But the sources emphasize osmosis. What's special about osmosis?
Osmosis is specifically about the movement of water. It's the passive diffusion of the solvent, which in biology is almost always water, across a semi-permeable membrane.
Semi-permeable meaning water can cross, but many dissolved things, solutes, cannot.
Exactly. And the rule is simple. Water moves from an area where water concentration is high, meaning fewer solutes dissolved in it, a dilute solution, to an area where water concentration is low, meaning more solutes dissolved in it, a concentrated solution. Water chases the solutes, trying to dilute them and equalize the concentration on both sides.
And this generates a force, osmotic pressure.
Yes. Osmotic pressure is technically the pressure you'd need to apply to stop the water from moving by osmosis. It's directly related to the concentration of solute particles. The more stuff dissolved, the higher the osmotic pressure, the stronger the pull on water.
And this isn't just abstract physics, it directly affects cell volume, right?
Critically, think about that red blood cell again. If you put it in pure water, hypotonic solution, water rushes in because the inside of the cell has more solutes. The cell swells up and can even burst hemolysis. If you put it in very salty water, hypertonic solution, water rushes out because the salt solution is more concentrated. The cell shrinks and shrivels crenation. Maintaining the right osmotic balance is essential for cell survival.
So water moves passively via osmosis. What about bigger molecules that can't just diffuse, but the cell still wants to move them passively? Like glucose, if it's more concentrated outside.
That's where facilitated diffusion comes in. Facilitated means helped. The molecule still moves down its concentration gradient from high to low, so no ATP is needed, but it can't cross the lipid bilayer alone. It needs help from a specific protein channel or carrier in the membrane.
Like a revolving door that only lets glucose through, but only spins downhill.
Good analogy. It binds to the carrier protein, the protein changes shape, and releases the glucose on the other side. Still passive, still driven by the gradient, but requires a specific protein helper.
And the sources point out some common features for any transport that uses these protein carriers, whether it's facilitated diffusion or the active kind we'll get to next.
Yes, these are important characteristics. First, specificity. A glucose transporter usually only transports glucose, or maybe very similar sugars. It won't transport amino acids. Second, saturability. There's a limited number of these carrier proteins in the membrane. If the concentration of the substance gets really high, eventually all the carriers will be busy. The transport rate hits a maximum, T8C, and it can't go any faster even if you add more substance.
Like all the checkout lanes being open and busy at the supermarket.
Exactly. And third, competition. If two similar molecules use the same carrier protein, they'll compete for binding sites. The presence of one can slow down the transport of the other. This is really important in pharmacology, designing drugs that might compete with natural substances for transport.
Okay, specificity, saturability, competition. Now for the hard work. Active transport. This is moving stuff against the flow, uphill.
Right. This is when the cell needs to move a substance from where it's less concentrated to where it's already more concentrated, or moving ions against their electrochemical gradient. This requires energy, usually supplied directly by breaking down ATP. The transport proteins involved here act as pumps.
They're forcing things where they don't naturally want to go. Figure 8 in the sources shows this. You see ATP being used by that transporter protein labeled 2. And we keep coming back to the prime example.
The sodium-potassium pump, TextVec plus dollar pump. It's absolutely fundamental, found in pretty much all animal cells. It's an active transport protein, an enzyme really, an ATPase because it breaks down ATP, and it performs that crucial continuous exchange
3 sodium ions kicked out
for every 2 potassium ions pulled in.
against their respective concentration gradients, because there's usually much more sodium outside and much more potassium inside.
Precisely. It maintains those gradients. This pump is constantly working, burning ATP to keep sodium high outside and potassium high inside.
And that specific 3 out, 2 in ratio does something else too, right? It makes the inside more negative.
Yes. Because it's pumping out 3 positive charges for every 2 it brings in, there's a net loss of positive charge from the inside with each cycle. This electrogenic effect is a major contributor to the negative resting membrane potential we'll discuss shortly. It's essential for nerve function, muscle contraction, even maintaining cell volume.
Its importance is just immense, underscored by that huge energy budget it consumes.
Absolutely. If cyanide stops ATP production, this pump fails almost immediately, the gradients dissipate, and the cell can't function. Dead in the water, essentially.
Okay, active pumps move small ions. What about really big stuff, like engulfing a bacterium or releasing hormones?
That involves major membrane remodeling, vesicular transport. This isn't about individual molecules crossing the membrane, but about wrapping large amounts of material in membrane sacs called vesicles.
2 main types, in and out.
Basically, yes. Endocytosis is bringing stuff in. The membrane folds inwards, engulfing material from the outside, and pinches off to form a vesicle inside the cell. If it's taking in large solid particles, like a white blood cell eating a bacterium, it's called phagocytosis, cell eating. If it's taking in droplets of extracellular fluid with dissolved molecules, it's pinocytosis, cell drinking.
And the reverse, getting rid of stuff.
That's exocytosis, a vesicle inside the cell, often coming from the Golgi and filled with waste products or manufactured substances like hormones or neurotransmitters, moves to the cell surface, fuses with the plasma membrane, and releases its contents outside.
So endo is in, exo is out, uses membrane, moves bulk.
Got it. Now let's pivot from transport to some special functional properties cells can have. The sources highlight contractility and secretory activity.
Contractility is the hallmark of muscle cells. It's their specialized ability to convert the chemical energy stored in ATP into directed mechanical energy, force, and movement. This relies on those myofibrils we mentioned, the actin and myosin proteins sliding past each other. And the sources mention a specific type, isometric contraction. What's that?
Isometric means same length. This is when a muscle generates tension, it actively contracts, but its overall length doesn't change.
How can it contract but not shorten?
Because the force generated is exactly balancing an opposing force or load. Think about just holding a heavy weight steady in your hand. Your bicep is contracting powerfully, burning lots of ATP to maintain that tension, but your elbow angle isn't changing, the muscle length isn't changing.
Like pushing against an immovable wall. We're just standing upright.
Exactly. Maintaining posture involves constant isometric contractions in your back and leg muscles to counteract gravity. You're not moving in the obvious sense, but your muscles are working incredibly hard.
Okay, that makes sense. And the other special property, secretory activity.
This is the specialization of cells, often grouped into glands, to produce specific substances and export them. The key distinction here is where they secrete it.
Endocrine versus exocrine.
Right. Endocrine secretion means releasing the substance, typically hormones, directly into the internal environment, the bloodstream or the fluid surrounding cells, to travel throughout the body and act on distant target cells. Think thyroid hormone or insulin.
And exocrine.
Exocrine secretion means releasing the substance onto a body surface or into a cavity that opens to the outside, usually via a duct. Think sweat glands releasing sweat onto your skin, salivary glands releasing saliva into your mouth, or pancreas releasing digestive enzymes into the small intestine.
Into the blood versus onto a surface. Clear distinction. This leads us perfectly into the most dynamic properties. Excitability and conductibility. How cells, especially nerve and muscle cells, handle electrical signals, the nerve impulse.
This is where it gets really exciting, electrically speaking. It all hinges on the membrane potential. We've touched on this, the fact that there's an electrical difference, a voltage across the cell membrane.
Because of the unequal distribution of ions, largely thanks to the Na plus K plus pump.
Precisely. Even when the cell is just sitting there, not being stimulated, it maintains this voltage difference. This is called the resting potential.
How negative are we talking? What do the sources say?
For typical nerve and muscle cells, the resting potential is usually somewhere in the range of negative 65 millivolts, MV, to negative 85 millivolt. The inside is negative relative to the outside. The cell is said to be polarized.
Okay, so polarized means charged up, ready to go, like a little battery.
Exactly like a little battery, holding potential energy. It's maintained primarily by the Na plus K plus pump constantly working, and also by the fact that the membrane is leakier to potassium than to sodium at rest, letting some positive potassium ions leak out, further contributing to the internal negativity.
So that's the ready state. The actual signal, the message, is the action potential. The sources call it an all-or-nothing event.
All or nothing is key. If a stimulus is strong enough to push the membrane potential up from resting to a certain critical level, the threshold potential, often around negative 55 milliV, then the cell fires. It generates a full-blown action potential. If the stimulus is too weak, subthreshold, nothing happens. There's no half-action potential. It either fires completely or not at all.
And this action potential is a rapid, temporary flip in the voltage.
A dramatic, very fast reversal of the membrane potential. Let's trace it using the information from figures 9 and 10. Once threshold is reached, we enter the ascending phase, also called depolarization.
Depolarization, losing the polarization, heading toward zero and beyond. What causes this spike?
Specialized proteins called voltage-gated sodium channels snap open. Remember, sodium plus piled up outside and the inside is negative. So when these channels open, sodium ions rush into the cell down both their concentration and electrical gradients. It's a flood of positive charge coming in.
And that influx of positive NAT plus overwhelms the resting negativity.
Completely. It causes the membrane potential to shoot upwards rapidly, passing 0 milliV and typically reaching a peak around plus 30 milliV or even plus 40 milliV. The inside of the cell actually becomes positive relative to the outside for a brief moment. That's the peak of the action potential.
The cell has fired its signal, but it can't stay like that. It needs to reset, the descending phase or repolarization.
Just as quickly as it rose, it falls. Two things happen almost simultaneously at the peak. First, the voltage-gated sodium channels automatically slam shut and enter an inactivated state. Sodium influx stops. Second, different channels. The voltage-gated potassium channels open fully.
And potassium K+ is concentrated inside.
Right, so now with the inside positive and potassium channels open, potassium ions rush out of the cell, taking their positive charge with them. This rapid efflux of positive charge quickly brings the membrane potential back down towards the negative resting level.
Repolarizing the membrane. Figure 9 shows it might even dip slightly below resting briefly.
That's called hyperpolarization, or the undershoot. It happens because the potassium channels are a bit slow to close, so potassium continues to leave for a short time, making the inside transiently even more negative than usual resting potential. But then they close, the Na plus K plus pump cleans things up, and the resting potential is fully restored.
But during this whole process, the cell isn't immediately ready to fire again, the refractory period.
Crucial concept. This is a brief period after an action potential during which the membrane is less excitable or completely unexcitable. It ensures that action potentials travel in one direction down an axon and limits the firing frequency. It has two parts.
Absolute and relative.
Yes. The absolute refractory period corresponds roughly to the depolarization and most of the repolarization phase. During this time, the voltage-gated sodium channels are either already open or are in that inactivated state we mentioned. They cannot be reopened by another stimulus, no matter how strong. Another action potential is simply impossible.
Okay, absolutely impossible for a short time. Then comes the relative refractory period.
This follows the absolute period, often during the hyperpolarization phase. Now, the sodium channels have reset to their closed but ready state, but the membrane is still hypopolarized because the potassium channels are still open or slow to close. An action potential can be triggered now, but it requires a stimulus that is stronger than the usual threshold stimulus to overcome the increased negativity.
So harder to fire, but not impossible.
Yeah.
And the AP generated might be smaller.
Yes. The sources mention the resulting AP might have a smaller amplitude and rise more slowly during the relative refractory period.
So this whole cycle, resting potential, threshold stimulus, rapid sodium influx depolarization, sodium channels inactivate, potassium efflux repolarization, brief refractory period, then back to rest. This is the nervous impulse,
Yeah.
or the muscular impulse.
That is precisely it. This electrical event, this action potential, is the fundamental signal used by nerve cells to communicate over long distances and by muscle cells to initiate contraction. It propagates along the cell membrane like a wave.
How does it move? Does the whole membrane fire at once?
No, that's the beauty of it. The action potential generated at one point on the membrane creates local electrical currents that depolarize the adjacent patch of membrane to its threshold. This triggers an action potential there, which then triggers the next patch, and so on. It's a self-regenerating wave that sweeps down the nerve axon or muscle fiber membrane without losing strength.
Wow. So from just managing ion concentrations with a pump, the cell creates this incredibly fast, reliable, long-distance signaling
Where
mechanism.
it's truly elegant. Think about it. The simple act of you deciding to lift your finger involves potentially billions of these action potentials firing in precise sequences across networks of neurons in your brain, down your spinal cord, out to the nerve controlling the muscle, triggering action potentials in the muscle cells themselves.
Which finally leads to those myofibrils contracting. It all connects back. We've really journeyed from the basic structure
The membrane border guard, the nucleus blueprint, the mitochondria power plants.
all the way to these incredibly dynamic processes. Active transport fighting gradients, proteins building and sorting, and culminating in this electrical signaling based on ion flows. The cell isn't static at all.
Not even for a microsecond. It's constantly active, regulating, responding. A buzzing hive of controlled activity, always maintaining that delicate balance between its internal life and the demands of the external environment.
And that constant activity needs constant energy. The sources mention ATP, but also PC, phosphocreatine.
Yes. Phosphocreatine, PC or CP, acts like a very short-term, high-energy phosphate reserve, especially in muscle and brain cells. ATP is the direct currency, but you only have a few seconds worth stored. When activity suddenly ramps up, PC can quickly donate its phosphate group to ADP to regenerate ATP almost instantly.
A buffer system for ATP.
Exactly. It buys the cell a little time for the slower processes, like breaking down glucose or fats via the mitochondria, to catch up with the immediate energy demand. It highlights, again, that ceaseless energy turnover required just to keep things running, especially those vital pumps.
Which brings us perfectly to our final thought for you, the listener, to take away from this deep dive. We've hammered home the importance of that sodium-potassium pump.
That 3 plus out, 2K plus in machine, burning maybe a third of your cellular energy just to maintain the ion gradients that underpin the resting potential, which is the foundation for every nerve signal, every heartbeat, every thought.
So consider this. If something disrupts that pump's function across your whole body, maybe severe dehydration throwing off electrolyte balance, or certain toxins, what happens when that delicate 3-to-2 ratio is compromised?
Where would you expect to see the effects first? Which systems rely most immediately and critically on that precise electrical potential difference that the pump maintains?
We think of heart function or brain activity as high level, but they are utterly dependent on this microscopic, energy-guzzling pump working flawlessly in billions of individual cells. How does knowing this deep cellular dependence change how you think about overall health, about things like hydration or electrolyte balance and their impact on major physiological functions?
Something profound to consider. The biggest systems rely on the smallest machines working perfectly. Thank you for joining us on this deep dive into the cell.